today an FDA approval in bladder cancer, a breakthrough therapy designation in diffuse large B-cell lymphoma, and ODAC hearing scheduled for a drug in HER2-positive breast cancer, and FDA elimination for REMS certification prior to select treatments, and 2 disappointing phase 3 results in non-small cell lung cancer and triple-negative breast cancer. Welcome to OncLive News Network. I am Dr. George Simon of the University of Texas MD Anderson Cancer Center. In urothelial carcinoma, the FDA has granted an accelerated approval to atezolizumab as a frontline treatment for cisplatin-ineligible patients with locally advanced or metastatic disease. This approval is based on findings from the phase 2 IMvigor 210 trial, in which the objective response rate with the PD-L1 inhibitor was 23.5%, including a 6.7% complete response rate. Atezolizumab previously received an accelerated approval as treatment for patients with locally advanced metastatic urothelial cancer whose disease progressed during or after platinum-based chemotherapy. This was based on a separate cohort from the IMvigor 210 study. These accelerated approvals of atezolizumab are contingent on results from the ongoing confirmatory phase 3 IMvigor 211 study, which is comparing atezolizumab with chemotherapy in patients with locally advanced or metastatic urothelial bladder cancer in the frontline setting and after progression on at least one prior platinum-containing regimen. The FDA has granted a breakthrough therapy designation to Tisagenlecleucel-T, also known as CTL019, for use as a treatment for adult patients with relapsed refractory diffuse large B-cell lymphoma after the failure of at least 2 prior therapies. The designation is based on findings from the multi-center phase 2 study, the Juliet study. Novartis, the developer of the CAR T-cell therapy, intends to present the study results at an upcoming medical meeting. In March 2016, Tisagenlecleucel-T was granted a priority review designation for the treatment of pediatric and young adult patients with relapsed refractory B-cell acute lymphoblastic leukemia. Under the Breakthrough Therapy Designation Program, the FDA will be more accessible in order to provide advice on the design and conduct of the clinical development program. In HER2-positive breast cancer, the FDA has scheduled an Oncology Drugs Advisory Committee hearing for May 24, 2017 to discuss a new drug application for neratinib as treatment for patients following prior treatment with post-operative trastuzumab. The FDA accepted the application on September 20, 2016. The application is based on a phase 3 results from the ExteNet study as well as the phase 2 control trial. ODAC will review the efficacy and safety data and make a non-binding recommendation on whether FDA should approve the drug. While the NDA was originally anticipated in the first quarter of 2016, the submission was delayed following a series of meetings with the FDA in which the agency requested a new statistical analysis of data from the ExteNet trial. For the new analysis, data were censored for patients who missed 2 or more scheduled disease assessments prior to recurrence or death. Regarding the initial ExteNet data, neratinib was found to demonstrate a 2-year disease-free survival rate of 93.9% versus 91.6% in the placebo arm. With the new statistical model, the 2-year invasive DFS rate were 94.2% and 91.9% for neratinib and placebo respectively. The results represented a 34% reduction in the risk of disease recurrence or death. The FDA has also eliminated the need for risk evaluation and mitigation strategy certification prior to the administration of erythropoiesis-stimulating agents for anemia due to myelosuppressive chemotherapy. The decision specifically applies to epoetin alpha and darbepoetin alpha. Epoetin alpha was approved in 1989 for patients with anemia associated with chronic renal failure, while darbepoetin alpha was initially approved in 2001 with inclusion of chemotherapy indication in 2006. In the trials leading to the approvals for the ESAs, it was determined that treatment with the agents led to a shorter overall survival and or increased risk of progression or recurrence in patients with breast cancer, non-small cell lung cancer, head and neck cancer, lymphoid cancers, and cervical cancers. Based on these data, a REMS was approved in 2010 and fully implemented in 2011 for both medications to ensure that the benefits of the therapies outweighed the risk. Amgen, the company marketing darbepoetin alpha, submitted data to remove the REMS, which were based on surveys showing that the prescribers were aware of the risks associated with ESAs and that the agents were being used appropriately and according to the approved indication. The findings showed a decrease in the number of patients receiving chemotherapy who warranted ESAs, showing a lower overall risk. The trend analysis also showed that the introduction of the REMS had little impact on ESA usage trends. The label for both agents specifically that they should only be used with myelosuppressive chemotherapy when at least 2 months of treatment with the chemotherapy are planned. Additionally, the agents are not meant to be used in combination with hormonal agents, biological products, or radiotherapy unless the patient is also receiving myelosuppressive chemotherapy. Finally, 2 phase 3 trials investigating the oral PARP inhibitor veliparib did not meet their primary endpoints. The studies evaluated veliparib in combination with the chemotherapy regimen carboplatin and paclitaxel in patients with squamous non-small cell lung cancer and triple-negative breast cancer. Full results will be presented at upcoming medical meetings or published in a peer-reviewed journal according to AbbVie, the developer of the agent. In the non-small cell lung cancer study, researchers evaluated the efficacy and safety of veliparib plus carboplatin and paclitaxel versus placebo with carboplatin and paclitaxel in patients with previously untreated metastatic or advanced squamous disease. Patients were evaluated based on their smoking status. The primary endpoint was improvement in overall survival in a cohort of patients who had smoked within the past 12 months and had more than 100 smoking events in their lifetime. In the TNBC trial, the addition of veliparib to carboplatin and standard neoadjuvant paclitaxel was investigated for the treatment of patients with early stage disease. Patients were randomized to 3 arms and treated with either a regimen of veliparib combined with carboplatin and paclitaxel, placebo plus carboplatin and paclitaxel, or placebo combined with paclitaxel. All regimens were followed by doxorubicin plus cyclophosphamide. The primary endpoint of the study was complete pathologic response. That's all for today. Thank you for watching OncLife News Network. I am Dr. George Simon.